try to make a quick and dirty, and we may have to restart with this next time, quick and dirty generic eukaryotic gene. It's also in, of course, our lecture manual, but I'm going to do this again. So we'll make a little generic eukaryotic gene. Of course, there's a lot of differences, a lot of variations on theme and stuff, but let's do this. Now, once again, as a continuous sequence of DNA, the boxes represent generally important kind of things. Okay, nice simple gene with just 3 exons here. Okay, we're continuing. Okay, so this is in effect the gene problem. Not surprising, during RNA processing, something we don't see so much in prokaryotes, in RNA processing, we're going to get rid of the intron sequences and glue the exons, or at least some of the exons, together to make the final messenger RNA. So this is our gene. Now, in front of the gene, of course, we're going to have short distance in front of it, an overlapping sequence that is sometimes called the cap site, because we will attach a modified nucleotide called a cap to the RNA that's being produced from this. And it also overlaps with the site of transcription start. Transcription, especially if I can spell the word right. And we're talking just a few bases, handful of bases away from the first thing. And here's going to be our initiation codon. In other words, it's the first, this is going to, in the RNA form, this is going to be the AUG codon, that is the beginning, marks the beginning of the gene. And over here we will have the DNA equivalent of the stop codon. That says the protein is over and done with. Okay, now that's about 5, 10 bases away. Now, over here we're going to have our promoter. And the vast majority of promoters have a sequence called TATA and a few other things. It's often called a TATA box. Not all promoters have TATA boxes, but most of them do. The promoter in prokaryotes binds the polymerase directly. It's not quite the case here. The promoters, the TATA box binds a very important basal transcription factor that's a cornerstone of the whole basal complex. A couple more proteins stick on, and only then do you bind the polymerase. The polymerase is held down more by protein-to-protein interactions than by the polymerase actually binding the DNA sequence. It does bind the DNA sequence, but don't forget, eukaryotic promoters are wimps. They are very weak. The polymerase will not be able to bind to that promoter and hold on without all the assistance. So the promoter binds polymerase and basal transcription factors. And you need all of those guys to do this. Okay, now, generally you'll have various distances from promoters. You will have, and this ranges anywhere from, yes?
What's attached to the exon 1?
Exon 1. Oh, nothing.
No, what is in the front?
Oh, in the front. Cap site, it's a little bit in front.
No, the other one.
Not that.
Oh, initiation codon. This would be a sequence for the initiation codon. In other words, I'm saying this is going to code for the very first amino acid we put on the protein. And likewise down here, the stop codon, the code for the stop codon that tells the ribosome protein is over, don't add any more amino acids, you're done. It's like the period at the end of the sentence. And this is the capital letter at the beginning of the sentence. Okay, all right, now what we have here is a battery of enhancers. They often call these upstream regulatory elements, UREs for short. A number of sequences, many times these things do overlap with each other too, by the way. They're going to range in distance from a few tens of nucleic bases away from the promoter to hundreds, and then some far more further away up to a couple thousand are going to be additional sequences. All of these things do is they bind specific transcription factors. Often they are capable of binding more than one transcription factor, so they will compete. A transcription factor, two transcription factors may have to compete for the same enhancer. So they have to duke it out. And concentration makes a big difference. If I'm one kind of transcription factor, and there's 5 of me, and there's only 1 of you, and we're going to both try to get to the same enhancer sequence, guess who's going to win, folks? Sorry, don't put money on you. Smart money does not go on you. Okay, on the other hand, if there's 20 of you, and you're going for the same one that the 2 of us are, I think we're both up the creek. Okay, that can happen. And here again, these sequences can overlap each other. So if something binds here, it prevents anything from binding here. Additional layers of complexity. And we can talk anywhere from tens to hundreds. For the close-in ones, a base pairs BP, not British Petroleum. And these can be hundreds to thousands. It occasionally will even find enhancers behind the gene, and sometimes you can find enhancers inside introns. That's not all that common, but that has been known to happen. So you can find these things all over the place. Most of the enhancer sequences are located upstream or in front of the gene. But you can find them in other places, such as inside introns. It's not a very common thing, but it can occur. Okay, so now we continue down past the last exon. Now what's going to happen, and I'm just going to, without putting in the sequences, I'm going to say the initial transcript of RNA, the first RNA, it's going to start at that transcription start site. We're going to continue it down. Obviously, as you go through, it'll have all the intron and exon sequences. It will continue past, and now we're going to have a few other sites of DNA. We have generally a few hundred base pairs away from beyond the stop codon. There's a site on the DNA that is sometimes called the polyadenylation site. It's going to be on this RNA as well, and that calls an enzyme complex, at least in the polymerase II genes, the messenger RNAs, it calls an enzyme complex that builds the poly-A tail that we find on all messenger RNAs. You don't find poly, that's a string of adenosines, a couple hundred adenosine bases, adenine bases, that's unique to messenger RNAs. Other RNAs don't have it. But if you're looking at polymerase II thing, this is what it is. It's a protein coding gene. This is a polymerase II future messenger RNA. The polyadenylation site is there. Now, we're still reading and making this RNA transcript. Somewhere down the line, you got to tell the polymerase, your job's done, stop, get off. So, somewhere down the way, we will have one or more, generally at least a few, sequences that are called termination sequence. The terminator. Okay. Okay, now in this case we have 3 termination sequences. What these do is basically cause the polymerase to fall off. Now they're not really good at it. So sometimes, like once in a while, the polymerase will get to this termination sequence and it drops off, and that's the end for the messenger RNA. But sometimes it'll read right through it. And then maybe the second one will get it. So Terminator 1 doesn't stop it, Terminator 2 stops it. And then, if that still doesn't work, maybe Terminator 3 end of the polymerase machine will finally push it off. So you will get your original transcript may vary in length depending on whether you read through how many of those termination sequences you've read through. Okay, well, that is a quick and dirty overview of our typical eukaryotic polymerase II gene. Of course, we have this kind of stuff in here too. We've got this in page 34 of the new one. So, we covered that. What we're going to look at when we come back on Thursday, what we're going to look at first is chromatin structure, basics of chromatin structure, the nucleosomes, and how the nucleosomes are regulated. Because we have to get the nucleosomes off of things like promoters and enhancers and stuff like that in order for transcription to occur. And we'll look at a couple of neat types of experiments that are often used to identify changes in chromatin structure and availability of this sequence or that sequence and things like that. So we'll take a look at those kinds of things later on. So first thing in eukaryotes, you've got to do something to that chromatin, got to do something to those nucleosomes in order for transcription to occur. So with that, I will see you folks, like I say, in like way outside, because I have to set those things up. It'll take me a few minutes to do.